Today we're going to look at the reproductive system. We're going to break this into two separate videos for the male and the female system. We'll do the male here in the first part. So the reproductive system is important for humans to be able to carry out sexual reproduction. Sexual reproduction is going to involve producing your offspring using your germ cells or gametes. So those would be the sperm and the secondary oocyte. So fertilization is going to produce one cell with one set of chromosomes that are going to be a combination from the two parents. So the reproductive organs are grouped as gonads. So we refer to those as the organs that are producing the gametes, and they will also secrete some hormones. There's going to be ducts involved with this that are going to transport, receive, and store the gametes, and then some accessory glands in there that are going to help to produce support materials. Gynecology is going to be the field of medicine that is going to deal with the female reproductive system, where in the males it's going to be urology. It's also going to include the urinary system for males as well in there, and so we don't have a separate field that is specific reproductive health for males. So in the males, the gonads are the testes where sperm are produced as well as hormones secreted from. There's going to be a system of ducts that are going to transport and store the sperm. This is also going to be where they mature, and then it will help carry them to the exterior of the body. These are going to include the epididymis, the ductus deferens, the ejaculatory ducts, and the urethra. There's also several accessory glands that are going to help add secretions into the semen. These include the seminal vesicles, the prostate, and bulbourethral glands. And then we have the other supporting structures like the scrotum, which is going to be the sac that supports the testes, and the penis that's going to be used to deliver the sperm into the female reproductive tract. So when we look at semen, it's going to contain the sperm plus other glandular secretions. So here we have a cross section showing the major structures. You've got the scrotum here with the testes, the penis. You can see that you've got a series of tubes here. And so for the male, you've got the urethra here, the bladder, and you can also see that the urethra is going to be a shared tube for both urine and semen in the male. And then we have a cross section here from a preserved human specimen. So the scrotum is actually a cutaneous outpouching of the abdomen that will support the testes. On the outside, you've got the raphe, which is the external median ridge. And then the scrotal septum is going to internally divide the scrotum into two sacs. So each of those will have a single testes inside of them. So it's made up of a subcutaneous layer and the dartos muscle. So associated with each testes, you have a Creamaster muscle. It's located in the Spermatic Cord. This is going to be the muscle that is going to pull the testes up to help them avoid cold. So when they are close to cold water or something like that, they're going to pull away to try and get warm. So during arousal, this is reversed by warmth. The reason that they are able to go up and down like this is normal sperm production requires a temperature that's 2 to 3 degrees below the core body temperature. So unless they're cold, they like to be hanging down a little bit further away from the body in order to stay cool. And so this picture is a nice one for showing the anatomy. You can see the raphe here, and then you've got the testes inside. You have blood vessels and all of the tubes here. You've got the spermatic cord, the Creamaster muscle that's going to go through where your inguinal canal is. So the testes are paired. They develop near the kidney, and then they're going to descend through the inguinal canal near the 7th fetal month. The tunica vaginalis is going to partially cover the testes. So with inguinal hernias, this is a bulge in the area of the inguinal canal where the tissue would protrude through. You have a weak spot in the muscle. This is a lot more common in males because of the testes making that trip through there. The Tunica Albuginea is internal to the Tunica Vaginalis. It's going to go inward. This is going to form the septa that divide the testes into lobules. So in each testes, you've got 200 or 300 lobules, and then those are going to have 1 to 3 seminiferous tubules where sperm is produced. So we've got a cross section here, and you can see where you've got the lobules. So the tunica vaginalis is going to be a piece of peritoneum that will descend with the testes into the scrotal sac. This is going to facilitate the movement of the testes within the scrotum. So in your seminiferous tubules, there are spermatic cells or spermatogenic cells that are going to form the sperm. The spermatogonia are the stem cells. They are going to come from primordial germ cells that arise in the yolk sac. They enter the testes during the 5th week of development. So this starts very early on. So you have your primary spermatocytes that are diploid, that will give rise to secondary spermatocytes that are haploid, and then the spermatids, and eventually the sperm. And those will go into the lumen. So your Sertoli cells or cestinacular cells are going to be support cells to help support this process. There's tight junctions in the blood-testes barrier. This is important because it's going to prevent having an immune response attack the sperm cells' surface antigens. We also need to nourish the spermatocytes, spermatids, and sperm. Excess sperm will be phagocytized, and then this also helps to control spermatogenic cells from moving too much. It will allow the release of sperm into the lumen. It's going to produce fluid for transport. There'll be secretion of inhibin. That will help to regulate the effects of testosterone. So inhibin, when you look at the name inhibits, it's going to inhibit testosterone. And it will also inhibit the FSH that stimulates the testosterone. So then we have the Latex cells. They're found in the interstitial spaces between the seminiferous tubules. They will secrete testosterone. So just to kind of review, mitosis is going to be the duplication of the chromosomes followed by 1 division that gives 2 daughter cells that are genetically identical. Meiosis is what we do with the gametes here. You're going to reduce the number of chromosomes with 1 duplication of the chromosomes, followed by 2 divisions. So that's how you end up with them being haploid. So this process begins with the spermatogonia, which are diploid. The stem cells undergo mitosis to replace themselves, and then some will continue their development. Your primary spermatocytes are diploid. Each of those duplicate their DNA to begin meiosis. So in meiosis 1, you have the homologous pairs line up. You have crossing over to give more genetic variation. The secondary spermatocytes are going to be haploid, and they're the 2 cells that are at the end of meiosis 1. So each chromosome is going to be made up of 2 chromatids that are attached at the centromere. So in meiosis 2, the chromatids separate. So in meiosis 1, homologous chromosomes separate. In meiosis 2, the sister chromatids separate. So your spermatids are the 4 haploid cells that you have at the end of meiosis 2, and they remain attached to each other by cytoplasmic bridges. Then eventually, with spermiogenesis, you have the development of those spermatids into sperm. So the spherical spermatids transform into the more elongated shape that we know goes along with the sperm. So we have the acrosome and flagella that are going to form. The mitochondria will multiply as well. So the Sertoli cells are going to help dispose of the excess cytoplasm. Spermiation is the release of the connections to the Sertoli cells. At this point, they're still not able to swim. So with the chromosomes, remember in a somatic cell, they're diploid. You've got 23 pairs of chromosomes for a total of 46. Each pair is homologous because they're going to have similar genes in the same order, but one member of each pair is from each parent. So we have 22 autosomes and one pair of sex chromosomes. The sperm are going to have the sex chromosomes in there, and they're either going to be an X or a Y chromosome. So for a female, they have 2 X chromosomes. Male have an X chromosome and then a Y chromosome. The Y chromosome is significantly smaller than the X chromosome. It mostly has genes for sperm production. So the gametes are haploid cells, and they have a single set of chromosomes for a total of 23 that undergo meiosis. So in meiosis 1, prophase 1, these stages are going to be similar to what you see with mitosis, but you have 2 sets of them for meiosis. Here the chromosomes become visible, you form a mitotic spindle, the nuclear membrane and nucleoli are going to disappear. So there are some events that are not seen in mitosis or meiosis 2 that are unique to meiosis 1. Synapsis, where you have all the copies of the homologous chromosomes pair off and they form these tetrads. When they're in the tetrads, this is where you're going to have the crossing over occur, where portions of the chromatids are exchanged between members of the tetrad to give more genetic variation. So parts of the maternal chromosome may be exchanged with the paternal one. So the new chromosomes that are made in the sperm would actually be they would have a combination of genes from both the man's mother and father. So this will lead to gametes that are unlike either of that man's parents. So here is just showing when you have prophase 1, your chromatids, the centromere. Here when you're getting ready for synapsis, and here having some crossing over occur. Here you can see where crossing over has occurred, that you've got some genetic recombination on the chromosomes. So in meiosis 1, in metaphase 1, your homologous pairs line up. In meiosis 1 and anaphase 1, those homologous chromatids are held together by the centromere that's going to get pulled apart to the opposite side. So they begin to separate in anaphase 1. And then in meiosis 1, telophase 1, and cytokinesis, it's similar to what you see with mitosis. You're going to have 2 cells that are going to have a haploid number of chromosomes here. So here we have metaphase. They're starting to separate in anaphase 1 and pulling apart for telophase 2. So in meiosis 2, we have those 4 phases again: prophase 2, metaphase 2, anaphase 2, and telophase 2, with similar steps. Your centromeres are going to split, the sister chromatids separate and move towards opposite poles of the cell. This way, each daughter cell produced by meiosis will give a net result of 4 genetically unique haploid cells or gametes. So we have prophase 2, them lining up for metaphase 2, them separating for anaphase 2, and making 2 more cells with telophase 2. So sperm formation, you have the seminiferous tubules that are going to have all the stages of sperm development. So to go from spermatogonia to primary spermatocyte, secondary spermatocyte, the spermatid, and spermatozoa. Those Sertoli cells are important to support the sperm production. The Leydig cells are going to secrete testosterone. So in here you can see this is a seminiferous tubule. This is the lumen inside. Here is where you're going to have those sperm cells being formed. And then you have the Latex cells in here that will produce the testosterone. And this shows the spermatogenic cells here going through this process of forming the sperm, how they start out spherical here as a stem cell, and then you can see the elongated sperm cell down at the bottom. This picture is going to illustrate what happens with your meiosis. So you have mitosis to maintain some of these stem cells in here, but then when you go through, you've got your diploid primary spermatocyte that's going to undergo meiosis 1 and then meiosis 2 to give you your 4 spermatozoa. This is showing here the process of spermiogenesis to give you a visual of how they go from being that spherical cell. In here you've got the spermatid nucleus, the centrioles. You start to have your microtubules and the flagella form. You're going to have the mitochondria. You're going to develop the acrosome on the tip, and the flagella is going to elongate so that you end up with your head, midpiece, and tail in your final sperm. So the acrosome is on the tip of the head. The neck is going to be where the head joins to the midpiece. That midpiece is going to have all of those mitochondria in there. So each day about 300 million sperm complete the process of spermatogenesis. So in the head you've got the 23 chromosomes because it's haploid. This acrosome has important enzymes for penetration. Down here you've got the tail, the neck. Where you join, you're going to have the microtubules form the remainder of the tail. So the principal piece is the longest portion of the tail, and then you've got the end piece, which is going to be a tapering portion. So after ejaculation, the sperm have about 48 hours that they can survive in the female reproductive tract. So different sources do give slightly different numbers for that. If you're looking and going, how long are these going to survive in a female, for looking at pregnancy concerns, you probably want to be a little more conservative and assume a little bit longer time than that, since different sources say different amounts of time, and the environment inside of the female is going to have some influence on that as well. So at puberty, we start to see the secretion of the gonadotropin-releasing hormone. That's going to stimulate the anterior pituitary to increase the secretion of luteinizing hormone and follicle-stimulating hormone. So luteinizing hormone, or LH, is going to stimulate those Latex cells to secrete testosterone. It's going to be synthesized from cholesterol, so it's a steroid hormone, and steroid hormones are derived from cholesterol. It's mainly in the testes, and it will suppress the secretion of LH and gonadotropin-releasing hormone via a negative feedback loop. So the enzyme 5-alpha reductase is going to convert testosterone into dihydroxytestosterone in the external genitals and prostate. So FSH is going to act indirectly on spermatogenesis. So it's going to stimulate, it's follicle stimulating hormone. It's going to act on the follicle. So both FSH and testosterone act on the Sertoli cells, and they'll stimulate the secretion of androgen binding protein that binds testosterone to keep the concentration high. Testosterone stimulates spermatogenesis, and then when you have adequate sperm in there, the Sertoli cells are going to release inhibin that will inhibit the FSH. So you've got a big negative feedback loop here going on with the testosterone providing a negative feedback loop here to shut down the gonadotropin releasing hormone that is stimulating the cycle. So androgens are masculinizing hormones. So you have some during prenatal development. Testosterone stimulates the male pattern of development or the reproductive system. It's going to stimulate development of the ducts and descent of the testes. Dihydroxytestosterone will stimulate the development of the external genitals. It will help with the development of male sexual characteristics. At puberty, it's going to bring about the development of the male sex organs and those secondary sexual characteristics. Also, it will play a role in the development of sexual function. So your androgens will contribute to male sexual behavior, spermatogenesis, and the sex drive. It also stimulates anabolism and protein synthesis, giving males heavier muscle and bone mass. So a lot of times people that do body work are able to even feel the difference pretty quickly in the density of tissue between a male and a female body because of having that extra bone and muscle mass. So here again we've got another image showing the testosterone production. So when you have a decrease in the level of testosterone, negative feedback loop can increase it. If it's too high, it will be able to shut it down. So the ducts of the testes, you're going to have pressure generated by fluid produced by the Sertoli cells. It's going to help to push the sperm along the seminiferous tubules into the straight tubules, the retitestes, the efferent ducts in the epididymis, and then the ductus epididymis. Your epididymis is a series of tightly coiled ducts. The stereocilia are microvilli that are going to help to reabsorb the degenerated sperm. So if the sperm do not leave, they do eventually degenerate. This is going to be where you have sperm maturation. They acquire their motility here and their ability to fertilize. The sperm can be stored in there for several months. And this will continue as the ductus deferens. It's also sometimes called the vas deferens. This is going to be what conveys the sperm during sexual arousement. And this will happen through peristaltic contractions. This area can also store sperm for several months. So a vasectomy involves cutting the ductus deferens to prevent sperm from entering the semen or leaving the body as a means of birth control. Because the sperm can be viable and stored in these tubes for several months, you can't assume immediately after having a vasectomy that there will be no sperm present. So they will tell a male that there needs to be several ejaculations that occur before they can assume that that semen is no longer going to have sperm in it. You don't want to assume right after the vasectomy you're good to go. So the spermatic cord is going to ascend out of the scrotum. It's going to have the ductus deferens as it ascends through the scrotum, the testicular artery, the vein that drains the testes and carries testosterone, autonomic nerves, lymphatic vessels, the Creamaster muscle. So your spermatic cord and ilioinguinal nerve are going to pass through the inguinal canal. The ejaculatory ducts, they're formed by the union of the duct from the seminal vesicle and the ampulla of the ductus deferens. They terminate in the prostatic urethra. They will eject the sperm in the seminal vesicle secretions just before the release of the semen into the urethra. So the final tube is the urethra, which is your shared terminal duct of the reproductive and urinary systems. So you've got the prostatic urethra and the membranous urethra, and then the spongy penile urethra. And where it ends is at the external urethral orifice. So here you can see the bladder. You can see here where you're going to have the penis. You've got the ejaculatory duct, the seminal vesicle duct, the deep muscles here, and this is the prostate gland that is going to surround that. So the prostate gland can become problematic for males in older age. You can see with the location in urethra going right through that. It's like a donut-shaped gland around the urethra, and it does continue to grow throughout a male's life. So eventually, if your donut puffs up too much, it can start to occlude the urethra and impair the flow of urine. So here we can see the urinary bladder, prostate gland in here. You've got your seminal vesicle, the ductus deferens. So the accessory glands are going to secrete most of the liquid portion of the semen. The seminal vesicles will produce about 60% of the semen volume. It's going to secrete an alkaline viscous fluid that is going to have fructose, prostaglandins, and clotting proteins. They're different than ones in blood. The fructose is to help feed the sperm. The prostate does about 25% of the semen volume. It secretes a milky, slightly acidic fluid that will have citric acid, several proteolytic enzymes, acid phosphatase, and seminal plasmin, which acts as an antibiotic to help protect the sperm a little bit. The bulbourethral glands, or Cowper's glands, they secrete an alkaline fluid that's going to protect passing sperm. It's going to neutralize acids from the urine in the urethra. So it's important to have all of these components in there to create a fluid that will help to support the sperm and protect them. And then there'll be mucus that lubricates the end of the penis and the lining of the urethra. So this is a similar picture to what you'd had before, just giving you three-dimensional perspective of things. Here you can see where you've got the prostate gland and the urethra coming up. And then we just have another view of it here, and some pictures from underneath the microscope. So these are your mucous glands, and cross-section slide of the prostate gland and the seminal vesicles. So semen is a mix of sperm and seminal fluid. Its typical volume is 2.5 to 5 milliliters, and it will typically have about 50 to 150 million sperm per milliliter. So there is a lot of sperm cells in there. It's slightly alkaline. The pH is 7.2 to 7.7, and that's because of those seminal vesicle secretions. It provides transport medium, nutrients, protection. It does take many sperm in order for one to be able to enter. So the semen will coagulate within about 5 minutes, and then it reliquefies in about 15 minutes due to the enzymes in there. So I'm not sure who decided to do that study and look at reliquefied semen, but somebody did. So when we look at semen analysis, it's important that they have forward motility. You want the ones that are able to swim straight. If they lack forward motility, it's not good for fertility, as well as having a low count or abnormal shaped semen is not good for fertility in there. So the penis contains the urethra, which is your passageway for ejaculation, as well as the excretion of urine. The body of the penis has 3 cylindrical masses of tissue in there with erectile tissue. There's 2 corpora cavernosum and 1 corpus spongiosum. So while they are relatively similar with the 2 corpora cavernosum, sometimes one is slightly longer or bigger than the other one. That can cause the erect penis to point slightly to one direction or the other. The glans penis is the terminal opening where you have the external urethral orifice. The prepuce or foreskin is going to cover the glands in an uncircumcised male. So the root of the penis is the attached portion that attaches to the torso. So an erection is going to involve parasympathetic fibers being released and causing local production of nitric oxide. That's going to cause the smooth muscle in the arterioles to relax and allow for dilation for large amounts of blood to enter the penis. This is the mechanism that drugs like Viagra work on, is they are going to promote vasodilation with nitric oxide. So here we just have some different examples of shapes of sperm on here and some different things that can happen with them. So that is the male reproductive system.